Alright guys, welcome back to Underground Science, and in this video we're going to be talking about homologous recombination, specifically relating to DNA repair. Alright, so first things first, we need to know what homologous chromosomes are and what sister chromatids are. Alright, so homologous chromosomes, so let's say you have, for example, right, chromosome 3 from your mom and chromosome 3 from your dad, right? They both have same genes, right? So like, for example, if this gene was for, I don't know, hair color, eye color, whatever, let's say eye color, right? They both are the same genes, meaning the gene over here on your, let's say the chromosome you got from your mom, is coding for eye color, and the gene over here, same place, right, on your chromosome that you got from your dad, same chromosome, let's say it's on chromosome 3, right, in this case, for example, this gene on your dad's chromosome, right, the chromosome you got from your dad, right, is also coding for eye color. But the only thing is, like, we'll go into this later in genetics, is that these genes can have, different alleles. So maybe this can be dominant, and then, this could be recessive or something like that, right? Which we'll go over later. But the main thing to know right now is that these are homologous chromosomes, meaning it's not this chromosome is from your mom and this chromosome is from your dad. It's the same chromosome number 3, but just like you'll have the same genes, but just different alleles, okay, different versions of that genes. Those are homologous chromosomes. Sister chromatids is when in the S phase of the cell cycle, you replicate your DNA, and then you get something like this, where you get, a replication of your chromosome here, and then same thing goes for this chromosome right here, you get a replication here. So I know it's pretty horrible drawing, but now this chromosome, so let's highlight it in different color and show it. This chromosome, right here, we got to replicate during the S phase. We had DNA replication, and then we got this chromosome right here. So these two chromosomes are sister chromatids. They're literally equal, identical, okay? Same thing here. Same thing goes for this side, okay? But you still have two number 3 chromosomes. So what we can do in homologous recombination is, it's going to be a double-strand repair, meaning we're going to use homologous recombination if we get a double-stranded DNA break, okay? And then we can use our sister chromatid. Let's say we have a gene here, and in this gene we have some sort of, whatever, a sequence of nucleotides, let's say a couple hundred, and then let's say for some reason we get a double-stranded break here, right? So what we can do is, we're literally gonna, since these are sister chromatids, we're gonna have identical, like, DNA on these sister chromatids. So we're also going to have that same sequence of nucleotides on the sister chromatid. So what we can do is, we can take the sister chromatid as a template and just, rebuild this part that got broken. So that's basically homologous recombination. But if the sister chromatid got damaged too, right, like in the same way that this sister chromatid got damaged, then we can use the homologous chromosome as a template, even though they're not going to be, fully equal to each other, because they're homologous chromosomes. But let's just say that we're using the sister chromatid for this video, and only one of the sister chromatids got damaged, all right? So let's say this is a gene sequence, right? Double-stranded DNA in one of the sister chromatids, and then so let's say this is chromosome 3 or something, for example, right? This is a sequence on one sister chromatid, this is the sequence on the other sister chromatid. They're identical, right? Because sister chromatids have the same DNA, because they're just, like, you basically went to the S phase of the cell cycle, just replicated the DNA. So you have two literally same sister chromatids, same DNA in your sister chromatids. So let's say we start here, and then we go to the next, and we get a double-stranded break, okay? So we got a double-stranded break. See, there's the break affected both strands, right, top and bottom. But the sister chromatid is intact. So we can use this sister chromatid, the same DNA sequence as here, as a template. So we literally, like, if let's say, for example, right, let's say this is a 3' to 5'. You can't really see it here, but 3' to 5', right? I can actually write that in or just move it over. So it's 3' to 5'. Okay? And then so what we can do is, if I take this strand, right? If we look at this 3' to 5' is exactly exactly complement, we should say, right, to this 5' to 3'. So if I take this broken strand, the 3' to 5' broken strand, and I use the 5' to 3' template, right, then I can just, okay, after here, see over here, starting here, the A, T, and then if I go on, we have A, right? So then the complement to A would be T, complement to A would be T, complement to T would be A, complement to T would be A, and then we continue, right, with our strand. Same thing, we can use the 5' to 3' strand that's broken and use the 3' to 5' as a template, right? These are just, ways that we could do it. And then we can use this. So the T is over here, so that means A would be over here. T is over here, so A would be over here. A is over here, so T would be here. A is over here, so T would be here. And then you keep on going. So basically you can form, new DNA with using the sister chromatid as a template, right? The sister chromatid as a template because it's the same DNA if it's a sister chromatid, right? So if we demonstrated that real quick, let's say we, recruited enzymes to rebuild this DNA, and we're using this sister chromatid sequence as a template. So if we're rebuilding the 3' to 5' strand, what we could do, right, is use the 5' to 3' strand on the sister chromatid as a template. So then, right, we could say, okay, well, we're going here. Well, which part is missing? So starting from here, right? So on the 5' to 3' sister chromatid is A. So that means on the 3' to 5', right, we have to have a T here. Then this is A, so we have a T here. Then we have a T here, so then this has to be an A here. T here, A here, right? So bam, we just rebuilt it because we use the sister chromatid as a template, right? So that's homologous recombination repair. Same thing, we go to this broken 5' to 3' strand, and we use the 3' to 5' strand on the sister chromatid that's intact as a template. So we go here, right? We have a T here, so that means we're going to have a A here, an A here. We have a T here, so that means we're going to have an A here. A here, we're going to have a T here, and then A here, we're going to have a T here, right? And this just keeps on going. So we just rebuilt this double-stranded break by using the sister chromatid as a template, okay? So that's homologous recombination, right, for DNA repair. And then next, what we can talk about is certain diseases relating to this, like, breast cancer, ovarian cancer, and Fanconi anemia and things like that. Actually thinking about that right now, what we should mention is, before we do the next video, is that to do this process of homologous recombination, something that we should know that's high yield is that there's two very important proteins that are needed in order to even do this homologous recombination DNA repair. Those two proteins are BRCA1 and BRCA2. Okay, so these are like the high yield proteins that we should keep in mind, because these proteins are very important for homologous recombination DNA repair. Okay, so that's it for this video, and I'll see you in the future videos where we'll talk about diseases relating to homologous recombination DNA repair. All right, so yeah, see you in the next video. Thanks for watching.